Okay, I was in here earlier and the dings were not going off, so I'm just going to assume that they aren't and then try not to be distracted if they come on in the middle of this. But anyways, I'm just going to start. So my name is Jessica Foley. I'm a PhD student at the University of Bristol, and I'm going to talk to you today about some of the results of my PhD work looking at the evolution of longevity in Heliconius butterflies. So I'm interested in the evolution of aging and lifespan. And across the animal kingdom, there's a huge diversity in lifespans, ranging from famously as little as a single day in the adult mayfly to over 500 years in a cold water mollusk. And studying naturally long-lived organisms can help us to find the solutions that evolution has found for the problem of aging. And most studies of senescence, which is kind of the physiological deterioration that accompanies age, have been conducted in mammals and birds. So there's been something of a call for more taxonomic diversity to better understand the spectrum of aging. And also, if you're interested in studying an organism across the lifespan, then it's a lot simpler to do that with organisms with lifespans on much smaller scales. So insects are emerging as really good candidates for the study of aging. And I study a genus of tropical butterflies called Heliconius, which are quite famous in evolutionary biology for their Müllerian mimicry. But a less well-studied aspect of their biology is their remarkable lifespans. So while most butterflies can only live for a few weeks, Heliconius have been known to live for almost up to a year. And what's really interesting about this lifespan extension is that it happened relatively recently in evolutionary time. So this figure on the left is a phylogeny of the Heliconini tribe, or at least all of the species for which there's available lifespan data. And you can see that the Heliconia species, which are shown in the yellow, have lifespans which far outstrip their other close relatives. And in fact, there's about a 25-fold difference between the maximum reported lifespan for the longest-lived Heliconius and the maximum lifespan for the shortest-lived Heliconini. And so obviously a big question here is how have they actually managed to achieve this extended longevity? And we think that a big part of this is linked to their evolution of a unique pollen-feeding behavior in Heliconius. So this is unique among butterflies. Most butterflies, including Heliconius' short-lived relatives, just feed on sugary nectar. But the evolution of a pollen-feeding behavior in Heliconius has provided them with a source of amino acids throughout their adult lifespan. And we think that it's part of this sort of nutritional enhancement that's allowed them to live for such a long time. And so this link was really conclusively explored in this great paper from the 70s that showed that was examining ovarian dynamics in Heliconius and showed that the amino acids from pollen allowed Heliconius females to continually produce eggs throughout the lifespan. And so the authors sort of put this in an evolutionary framework and say that once heliconines began collecting pollen, selection would then favor an increased lifespan. And the proposed evolutionary scenario for this goes something like this. So if you can consider the last common ancestor of Heliconius and the non-pollen-feeding Heliconini, it was likely a nectar feeder, so it's born with a finite number of eggs. After about 1 to 2 months, these deplete, it reaches the end of its reproductive lifespan, and it dies shortly thereafter. So longevity would not be selected for in this scenario because of this cap on reproductive output. And this is the case that we see with the extant non-pollen-feeding Heliconini, such as Dryas Julia. Whereas when you then add this pollen-feeding adaptation, this continual egg production means that any mechanisms that confer extended longevity will be selected for because any individuals living past this point will be contributing more to future generations. And this is what we see for the Heliconius, such as Heliconius secale. So I wanted to really sort of robustly interrogate this link between pollen feeding and lifespan in Heliconius. So is pollen feeding really the direct cause of their lifespan? Without it, do they live for just as short as their short-lived non-pollen feeding relatives? And also, would provision of pollen have any impact on the non-pollen feeders? So is there any kind of benefit to incidental amino acid uptake? That paper showed that pollen feeding helps to delay reproductive senescence in Heliconia, so I wanted to know if access to pollen might impact other aspects of senescence in these butterflies. And I also wanted to know if the senescence might differ between shorter and longer-lived genera. And then finally, once we sort of disentangled these evolutionary sort of bases of lifespan in Heliconius, I'm also interested in finding out what some of the more proximate mechanisms of this longevity might be. And so to ask these questions, I've leveraged the sort of lifespan differences across the Heliconini tribe and used a sort of comparative framework studying one representative long-lived pollen feeder, Heliconius secale, and one representative short-lived non-pollen feeder, Dryas Julia. And to assess the impact of pollen, I've conducted all these experiments under both a pollen-fed and a pollen-deprived treatment. So first up is my survival analysis. So on the x-axis, we have time in days, and on the y-axis, we have survival probability. And then the lines in orange are Dryas Julia, that shorter-lived non-pollen feeder, and the purple lines are Heliconia secale, that longer-lived pollen-feeding butterfly. And the dashed lines represent the pollen-deprived treatment, and these solid lines represent the pollen-fed treatment. So straight away, we can confirm that, yes, cicale live for much longer. And we can also see that access to pollen doesn't seem to have any impacts on dryas lifespan. However, pollen deprivation does seem to significantly reduce cicale lifespan, but critically not down to the level of their short-lived relatives, dryas. So there seems to be some kind of a plastic response to this, to the pollen, but there also seems to be other mechanisms at work. So we can see there's sort of a genetic effect, and there's an environmental effect, and there's also this sort of genes by environment effect. So we think that this is just, suggestive of underlying evolved heritable mechanisms of longevity. And we can take the survival analysis one step further. So while traditional survival analysis allows for dissection of total lifespan differences, fitting cumulative deaths to a parametric survival distribution allows you to understand more about the shape of mortality. So you get information about the probability of dying at a specific time. So to kind of explain this, I'm going to skip over a little bit of the maths, but basically, once again, on the x-axis, we have age in days, and on the y-axis, we have the natural log of the hazard, so sort of the probability of dying. And if you picture a hypothetical population, we can see that as it ages, it gets exponentially more likely to die. And an increase in the intercept of this line is indicative of a higher baseline mortality, or a higher mortality independent of age. So this suggests that this population across the lifespan is just more likely to die. And this is usually an indicator of an increased frailty or an increased sort of vulnerability to extrinsic hazards like trauma or starvation. Whereas an increase in the slope of this line is indicative of an increase in age-dependent mortality, so a higher rate of aging. And so what this kind of means is that that population is more quickly becoming more likely to die. And this is often reflective of an underlying physiological senescence. And this is really helpful because you can see that in both of these scenarios, lifespan is decreased, but for kind of different reasons. And so this helps you sort of dissect and explain observed differences in lifespan. So when we apply this kind of analysis to my survival data, we can see a couple of patterns start to jump out. So first of all, between the two species, there doesn't seem to be any difference in baseline mortality, but cicale seem to have evolved a slower rate of aging. And what this kind of means in real-world context is that while both these species are subject to the same extrinsic hazards early on in life and are equally as vulnerable to them, dryas seem to decline faster physiologically. And this is the explanation for the observed differences in lifespan that we see. And then when you look at the impact of diet, we can see that there's, as expected, no impact in dryas. But we can see that in cicale, this sort of reduced lifespan in pollen-deprived butterflies seems to be due to an increase in baseline mortality, not due to any kind of increase in the rate of aging. So we also wanted to find some kind of a physiological correlate of this. And we know that across a couple of different insects, including Drosophila, they report age-related declines in muscle fitness. And so we used a grip strength assay, which is essentially just how much weight a butterfly can pull with its true legs, to try and use this as a proxy for condition. And so we find a couple of sort of similar trends here. So once again, diet seems to have no impact on dryas grip strength. We have age on the x-axis, and then on the y-axis, grip strength is now measured in grams. So diet doesn't seem to have any impact on dryas grip strength, probably quite expected. And we can see that pollen-deprived cicale are actually weaker. But interestingly, this is consistent across the lifespan. And this is quite nice because it quite neatly mirrors what we've seen in our parametric survival analyses. So pollen-deprived cicale are weaker, and it makes sense that this might lead to an increased baseline mortality. That's a little bit intuitive. But also critically, pollen-deprived cicale don't seem to show any signs of aging any faster. These lines are still parallel. However, what's even more interesting, I think, is that dryas do actually seem to weaken with age. So we can see that by week 5, these butterflies are actually pulling on average about 16% less than they are on week 1. Whereas this doesn't seem to be true for cicale, which don't seem to show any signs of weakening with age. And this is true even when you extend the data across the full cicale lifespan. So even at the oldest ages, age is not a significant predictor of grip strength in these butterflies. And this is quite surprising considering the evidence for this in their close relatives and in other insects. And we think this suggests that in accordance with their extended lifespan, these butterflies have also evolved a delayed senescence. So we can see the pollen does seem to provide some direct benefits to heliconia's fitness. And critically, these benefits don't seem to be true for their close relatives, Dryas Julia. So heliconias have not only evolved the ability to actively collect and digest pollen, but also to exploit it for its nutritional advantages. And when I talk about those nutritional advantages, I've already mentioned the fact that the amino acids in pollen allow for continual egg production, but they're also used in repair and in energy storage. And pollen's also a source of lipids, which are important in insect immunity. And so we can see how some of these nutritional advantages might lead to some of the benefits to Heliconia's fitness that we've seen. So, for example, that reduced baseline mortality, and therefore their increased lifespan, and also in their improved performance on the grip strength assay. This might be reflective of their higher energy budget from the amino acids. And there's like some pretty clear ecological relevance for being stronger, so things like competition for resources or predator avoidance, which might then in turn be reflected in their reduced baseline mortality. But importantly, there doesn't seem to be any sign that feeding on pollen slows the aging process, and this is shown in the similar rates of aging as evidence from our parametric survival analysis, and also the fact that we don't seem to see any signs of physiological senescence in either of the two treatment groups. And I think a lot of this makes sense when you put it into a life history framework. So according to life history theory, increases in longevity come with corresponding trade-offs. But we think that the evolution of pollen feeding appears to have sort of loosened these life history constraints in these butterflies. So it's allowed for them to shift the burden of reproduction to the adult stage, and then evolve a shift in larval resource allocation towards somatic maintenance and longevity, so allowing them to stay robust for a lot longer. And there's quite a few potential proximate mechanisms for this. One suggestion is an increased investment in immune function. Another one is an increased investment in antioxidant defenses, which are pretty highly implicated in the aging process. And some preliminary results that I have suggest that this might actually be the case. So this is a graph showing levels of superoxide dismutase activity, which is a key antioxidant enzyme, and it appears to be a lot higher in Heliconia secale as compared with Dryas Julia. So this suggests that they might be actually increasing investment into these pathways. However, it's also important to remember that aging is a very broad, multifarious process, and there's probably many, many interacting mechanisms at work here. But that just means that there's, as ever, more research to do. So to conclude, we've shown that pollen does indeed provide direct fitness benefits to Heliconius in the form of a longer lifespan and also in the form of improved grip strength. But more importantly, we've shown that the lifespan extension in Heliconius is not solely due to just these plastic benefits of pollen. They've also evolved a lifespan extension facilitated by the slowed rate of aging that's independent of diet. And alongside this, they appear to have evolved a delayed physiological senescence. So we think this is indicative of evolved heritable mechanisms of longevity, and there also appears to be substantial variation within the genus in terms of lots of different aging metrics. So I think that all of this presents Heliconius as a really interesting testing ground for the study of the evolution of long life. And with that, I would like to thank my three interns who worked with me on this project, Josie, Maid, and Greta, as well as my primary supervisor at Bristol, Dr. Stephen Montgomery, and my collaborators, Dr. Owen McMillan and Dr. Pablo Buraco, and also all my funders. So yeah, I'd be happy to take any questions.
I forgot how something fit in. Earlier in the talk, you showed a graph that did have a slower rate of aging, and then by the end, we were concluding that that didn't have a slower rate of aging. Can you remind me what the first graph was of, or was it just hypothetical to show us what it was?
No, yeah, sorry. So that was the two different species. So yeah, sorry, maybe I didn't make that super clear, but there's two different species, and one species seems to have a slower rate of aging. But the thing that I was concluding was that it's not that the pollen slows their aging.
It's not.
Yeah. Yeah.
In many insects, the two sexes have very different dietary requirements. Your work seemed to be focused on females. Can you comment at all on the impact and the dimorphism in longevity?
Yeah, absolutely. So the work actually was done with both sexes. It's just that you would just expect there might be some kind of sex-specific effects because of this sort of hypothesis that it's facilitated by their prolonged reproductive lifespan in females. But I did all this with both males and females, and there actually weren't any sex differences. It wasn't a predictor of lifespan in either of these two species. And I've also run similar analyses on, I think, a total of 5 Helicanini species very specifically, but a couple of broader ones with some patchier lifespan data, and I haven't found any evidence for sex differences in lifespan. So yeah, maybe the pollen is also doing something for the males that's a little bit less easy to quantify than eggs. Yeah. Okay. Oh, sorry, there's one more in the back. I think I might have a minute.
About the pollen, do you know the composition of the pollen that you provided in amino acids and proteins?
So there has been some work done on it, and we have basically the helicones differ somewhat in levels of pollen reliance, but also in the specific plants that they prefer to collect pollen from. There has been some amino acid analysis of their sort of primary pollen sources, so Siguria plants. So there is a breakdown of the different amino acids, but I haven't done any deep dives personally into, varying levels of different amino acids and seeing what that might mean. But yeah, there is that data for people who know more about proteins than I do. Yeah. Okay, great. Thank you very much.
My talk is on the long-run evolution of aging. This is in the sense of long-run equilibria. I'm not trying to predict what's going to happen in a billion years to us. I'm trying to understand the way things age now in terms of they've been evolving for a long time and maybe they are in some kind of equilibrium state. What is aging? Aging is the thing that makes you get worse when you get older and have more birthdays, often called senescence, but for the purposes of a talk, I'm just calling it aging. You can see, for instance, humans age because we become progressively more likely to die within the next week or year as we get older. Not all species age. Freshwater hydra don't seem to. A 200-year-old hydra is just as likely to survive another week as a 20-year-old hydra is. So that's how you define what aging is. I'm not looking at specific molecular things. I'm looking at these demographic rates. Similarly, again, I'm not looking at the molecular causes of aging. I'm looking at evolutionary explanations of aging and how this fits in with demographic rates. And you will see this graph again later on the left. You might already be familiar with it. Here again is some of the diversity of age-structured life histories, which refers to any age-specific pattern of mortality and fecundity, whether or not you age. So 2 human populations, you can see that we age. So-age sheep also age. I believe the next talk is going to be specifically about soy sheep, so that's fun. Freshwater hydra don't age, constant fecundity, constant mortality. There are even things that seem to age negatively, getting less vulnerable as they get older and more fecund as they get older, like desert tortoises. I don't know if that holds forever, but at least for the observed age ranges, they seem to. So why do we age? Aging surely isn't good for us. It's surely not fit. And the answer is it's not fit. It's bad for us. But natural selection doesn't care that much because natural selection doesn't care that much about late life. If you imagine, if a mutation is going to make your heart explode and kill you, but it only does it when you're 80 and you've already had most of the kids you're ever going to have anyway, it doesn't really matter. So these are the people who figured that out. JBS Haldane appealed to this idea of talking about Huntington's disease, which is a mutant gene that kills you after you've already had a few kids because you're in your middle age. Medawar and George Williams turned this into an evolutionary theory of aging, or kind of 2 overlapping evolutionary theories of aging. The version associated with Medawar is the mutation accumulation theory, which just says there are genes that are silent early in life and bad for you late in life. Those cause aging. George Williams, and also separately Medawar, but it's more associated with Williams, said maybe it's actually genes that are good for you early in life and then bad for you late in life. Maybe that's what causes aging. And you can flesh these out in more mechanistic terms. You can turn, for instance, the damage-based disposable soma theory is kind of a version of antagonistic pleiotropy. So is the hyperfunction theory. Both of these are different ways to say, here's a kind of early benefit that might come with a late cost. The most important for this presentation, though, is Hamilton, who took this vague idea I've just conveyed to you and turned it into math. And it is challenging to show math theory stuff in a talk, but I will do my best by translating it all into English. So the force of selection declines with age. What is a force of selection? It is that partial derivative. If a mutant affects a trait and thereby affects fitness, you can divide this into its effect on the trait and how much fitness cares about that trait, or specifically how much it cares about small additive increases or decreases in that trait. If you've ever, drawn the slope of a line and multiplied by a thing, then you know what this is. So to flesh that out, you need to make some assumptions, which Hamilton made. He assumed that fitness is the intrinsic rate of increase R, which is even if a population that has age structure and overlapping generations has reached its stable age distribution, what's called, it'll grow exponentially, and R is the exponent.
And you can kind of think of the population of your own descendants as your fitness. And he also assumed that mutations additively increase the force of mortality, which is a measure of mortality that's not quite probability of death, but when it's low, it's similar, or fecundity. And these turn out to matter for the exact equations that he got, which are these. Aren't these easy to understand? No. So that's why here we go. So the force of selection on mortality with his assumptions turns out to equal, the T on the bottom is just generational length defined as the average age of mothers, so you can ignore that. So the force of selection on mortality is the fraction of newborns with mothers X plus 1 or older. Obviously, the higher X is, the fewer mothers can be older than that. But no mothers are younger than the age of maturity anyway, because that's what it is to be a mother. So the force of selection on mortality is constant until age of maturity, and then it starts to decrease. Similarly, selection on fecundity. In the stable age distribution, you might have like more adults than newborns or less or whatever. Usually you'll have less older things because creatures die off. So this one also tends to decrease with age. And here is a calculated example for the desert tortoise that we saw. Interestingly, even though the desert tortoise negatively ages, its forces of selection still go down with age. So that's what Hamilton did, but his assumptions aren't necessarily the only assumptions that you can make. Fitness could be lifetime reproductive output instead of the intrinsic rate of increase. That's a suitable assumption if the population is density dependent and held at a constant size. You could also have mutations, for instance, that multiply mortality rates instead of adding to them, or also for fecundity rates, but we'll talk about mortality. This would be called the proportional hazards assumption instead of an additive hazards assumption. And this makes a difference to which equations you get. Here's a table that you don't have to worry about. But interestingly, as a side note, some of these can actually sometimes increase with age. Not easily and not all the time, but sometimes. So it's pretty faint, but you can see there's a little bit where some of these dotted lines there actually go up a little bit, whereas the solid lines can't go up. So that's all background. Now we're going to get to the stuff that I'm actually trying to do. One of the limitations of this general framework completely is that all of these forces of selection are very near term. It's like if you hit a baseball and you calculate the force with which you hit it, that's not necessarily going to tell you how far the ball will go. You know, what's the friction in the air? Who knows? And the issue is that, and a complication is that the forces of selection are themselves functions of the life history. So force of selection is affecting how the life history evolves, but then as the life history evolves, the forces of selection also evolve. So if you're trying to understand where a life history is eventually going to end up, it's challenging to do. So how do we do this? One approach is simulation, but I'm not doing that. Another one is to try to just come up with some simple evolutionary models and see if you can derive some equilibria, see if you made them simple enough that they're actually analytically tractable. Or if you're a little bit lazier and less clever like me, you don't find the equilibria, but you find some conditions that an equilibria would have to satisfy to exist. So I come up with two simple models. One of them is a mutation accumulation model. One of them is a reproductive effort model. We'll do the mutation accumulation model first. So the idea here is that you have mutations with age-specific effects on some measure of mortality. We'll decide later if we're doing additive or proportional. And the mutations arise at the same rate for every age with the same distribution of effect sizes at every age. And then the reproductive, excuse me, sorry. And for the reproductive effort model, we're talking about the cost we're looking at, the cost of reproduction, is a same age cost of mortality, and it's specifically a multiplicative increase in mortality or an additive increase in log mortality. So these are the two models I'm looking at. For the mutation accumulation model, the intuition is that at every age, mutation pressure is pushing the mortality rate up, and selection pressure is pushing mortality rate down, and the equilibrium for a given age is where these cancel out. But then if the idea is that if every age is in equilibrium for mortality, and the mutation pressure is the same at every age, then the selection pressure also has to be the same at every age. So this is the idea of what we need to be the equilibrium condition for this model. Is it possible to have such an equilibrium? Not if mutations are additive hazards like Hamilton thought. Because if you remember, the thing Hamilton famously showed is that that force of selection has to decrease with age. It can't stay the same with age. So there is no equilibrium under this model for additive hazards. But for proportional hazards, there are. And the equilibrium turns out to be that mortality rate is inversely proportional to relative abundance in the stable age distribution times reproductive value at that same age. This is for a continuous time. If it's discrete time, it's slightly different. It's actually residual reproductive value, but that's okay. Interestingly, this also is another way to rewrite Hamilton's original mortality indicator. So interestingly, Hamilton's mortality indicator appears in the equation for the equilibrium condition for not the assumptions that Hamilton actually made, which can throw people off. And that's part of why I wrote it this way instead of writing it that way. So now to the reproductive effort model. The idea here is that no matter what else is happening, no matter how many trade-offs have happened, no matter how many mutations have happened, there is always one more trade you can make if you choose to or if it's fit to, which is that you can always additively increase your fecundity at the price of also multiplicatively increasing your mortality or additively increasing your log mortality. Again, the equilibrium for a specific age is where the fitness benefit equals the fitness cost, so they cancel out. That's the math version of that. And rearranging that thing you just saw gives you this equilibrium condition. So this time in this model, at equilibrium, mortality is not inverse to age distribution times reproductive value, it's just inverse to reproductive value. So to anyone who read ever or skimmed Ronald Fisher's 1930, The Genetical Nature, or well, the Genetical Theory of Natural Selection, he would be very excited by this because he remarked that, so in humans, reproductive value goes up until the age of maturity and then starts to go down, whereas mortality goes down until maturity, then goes up. He thought this couldn't be a coincidence, but he gave the wrong reason why. He thought reproductive value, I'll say what reproductive value is. Reproductive value is at a given age, it is your expected future reproductive output, but where farther future offspring are discounted according to population growth. So he thought reproductive value was the strength of natural selection, and that was wrong, and Hamilton corrected him on that. But interestingly, you still get this thing that impressed him in this model. So that's exciting. So here's the two models, here's the two equilibrium conditions. The first thing to notice is that they are different from each other. So whether or not either model is true, the fact that different models have different equilibria is itself interesting and in itself tells us something about, about evolution of aging. But so now to talk about some of the major feature or the widely discussed features of real life histories and whether these models have anything to say about it. High juvenile mortality. Babies die easily, then mortality decreases until maturity and then starts to go up in humans. The mutation accumulation model cannot explain high juvenile mortality at all because it's inverse to Hamilton's force of selection, and Hamilton's force of selection can never go up, which means in this model, mortality can never go down. On the other hand, the reproductive effort model can explain high juvenile mortality, and in fact, it requires high juvenile mortality because if you're in a juvenile and you're getting close to maturity, you're passing the hurdles that you have to pass to get to maturity, and you're getting closer to starting reproducing. So interestingly, that model actually requires high juvenile mortality, which is kind of neat, because high juvenile mortality is a common thing. Catastrophic senescence is the idea that in some mutation accumulation models that people study, this is theoretical, not an empirical thing, they often use additive hazards in their mutation accumulation models, and often they find that as mutations build increasing mortality, they also decrease selection against further increases in mortality, and the whole lifespan just erodes, and then the thing either goes extinct or becomes like really short-lived or whatever. This mutation accumulation model, I think, sheds light on why that's the case, because it's been seen in various models, but here it explains why. It's because there can't be a equilibrium with the additive hazards assumption, because if there was an equilibrium, that would mean Hamilton's indicator was staying constant with age, and Hamilton's indicator cannot stay constant with age. So I think this sheds light on a feature of those theoretical models. Let's see. Negative senescence, negative aging, mutation accumulation model can't handle it. Reproductive effort model can, because reproductive value can go up with age, which means mortality can go down with age. Post-reproductive lifespan, we live after we stop giving birth and so on. That's probably because we have parenting and grandparenting, so that's an adaptation. So the models, as I've described them here, can't handle that, because when we stop reproducing, the version of reproductive value I'm using goes to 0. You might be able to redefine reproductive value so it takes into account parenting and grandparenting, but I haven't done that. And there's a couple more in there, but I'm gonna skip it. But I think the most important thing about this exercise is that the equilibrium conditions are mutually exclusive. They cannot both be true at the same time, because CX, the relative abundance in the stable age distribution, the age distribution cannot be flat from 0 to infinity without absurdities. So that basically, for them to be equal, CX would have to be 1 always for every age, and that can't happen. Therefore, if the evolutionary processes that the two models are describing are both happening, then one or the other can't be in equilibrium, and that's interesting because we're used to often thinking, people who talk about these mathematical models of aging evolution often think of it as like a very, very a priori thing, like one of the few things you can discover about life by sitting in your armchair is, oh, lots of things will age, and that is true, but exactly how they age depends a lot on the kinds of mutations that are happening, the kinds of genetic variation that there is. And the genetic variation, it's not just affecting, how quickly you can approach an equilibrium, it's affecting what the equilibria are. And that itself, even though these are completely theoretical, completely silly models, that itself is interesting and important, I think. And, okay. Anyway, here's some very rough and dirty attempts to, compare it to real life, and who knows, useless. Cool.
Okay, hi everyone. I'm Lizzie. I'm just about to start the third year of my PhD at the Institute of Ecology and Evolution at the University of Edinburgh, and today I would like to talk to you about the effects of early life adversity in the Surrey sheep population. So we know that early life adversity has been reported to have long-term effects on physiological function, demographic rates, and lifetime fitness. Favorable conditions in early life can have positive consequences in later life. This is typically called the silver spoon effect, but also the reverse, that negative conditions in early life can have negative consequences in adult life. But an alternative hypothesis is sort of like a selective filter idea, where harsh early life conditions can select for the more robust individuals in a cohort. Most examples in the literature about the effects of early life adversity come from humans and primates, although there are some examples in wild populations, notably badgers and baboons and red deer. But most of these studies use a composite or cumulative measure of adversity or a small number of environmental variables. And although these studies are great, they might not be able to capture the complex and multivariate environments that are faced by wild populations. I'd also like to talk about how the sexes could possibly be differentially impacted by early life. Especially in polygynous systems, males tend to have greater requirements in early life due to rapid growth, larger body size, and the cost of secondary sexual characteristics. So we might expect them to be more sensitive to the harsh environmental conditions experienced during development in the neonatal period. So my study system is the Surrey sheep, which are found on this very tiny island quite far away from Scotland. And this is a wild, unmanaged population who've been studied for about the past 40 years. We have data on over 10,000 individuals. We catch the lambs shortly after birth, weigh them and tag them. And then about roughly 60% of the population is caught again every summer. Mortality in this system is usually over the winter period, and we know it's exacerbated by harsh weather and high parasite burden and high population size. This is a highly polygynous system, and it's also a very sexually dimorphic system, with the males being considerably larger than females, and about 85% of the males having these very large, very beautiful, big curly horns. So what aspects of the early life in this population have I been thinking about? So if you think about the first year of a lamb's life, there's several things that we record in this system. So we know the winter weather just before they're born, how much they weigh when they're born, if they have any siblings or not. So that's a twin born at the same time as them. We can work out the mean parasite burden within their cohort. We also know the population size at the end of the summer. We know the winter weather during their first winter, and we can also work out whether they're orphaned during their first year, at any point during their first year of life. So what am I defining as adverse? Like, what is bad in this population? So that would be whether the winter is harsh just before they're born, if they're born small, if they have a twin, if the parasite burden is high, or whether the population size is high, if there's a harsh winter weather in their first winter, and whether their mum dies. So under the silver spoon hypothesis, I expected that I would see reduced first-year survival, reduced lifespan, and reduced lifetime breeding success if an individual had experienced one of these conditions or a combination of them, and also that males would be more negatively impacted than females. Okay, so what did I find? So all of the conditions that I looked at, apart from weather just before they're born, all led to reduced first-year survival. And the plots that you can see with the multiple colours are where males and females differed. Males, the blue line males are, for these three, so parasite burden, population size, and first winter NAO, that if they experience these, they're much more likely to die than females. I just like to point out, NAO at this time is like a measure of atmospheric pressure across Europe. Low conditions are like cold and dry, and high values are sort of like wet and rainy, and they really hate wet and rainy. They're not big fans of that. So when I looked at adult lifespan, I found that only 2 of the measures of early life adversity affected lifespan. So birth weight and first winter NAO. So birth weight had the same effect for both males and females, but only males were affected by first winter NAO. But this isn't like the opposite of what I predicted. The ones that go through these really awful winters seem to be the ones that are living longer. And what we think is happening, if you think back to the plot on the last slide, is that males who are able to survive these really horrible winters are the like more robust individuals in the population. They're the really good ones. So they're much better at surviving winters than individuals that don't go through these conditions during early life. So when I look at lifetime breeding success, only 2 of these metrics didn't influence lifetime breeding success. So for birth weight's great whether you're a male or a female sheep. The effect is much more positive if you're a male, though. And for the other 2 where I found a difference between the sexes, females were not affected, but males were affected. So males that go through these really large population sizes in their first year of life tend to have reduced lifetime breeding success. And we think this is because of like reduced resource availability during their first year of life. Means that they're not able to grow as quickly, and because breeding success in this population, at least for males, is really determined on size. Males that are not able to get as big quickly sort of lose out in the first few years of their life before they catch up. There was, I can't show this plot here right now, but there was no effect of first winter NAO for males on their annual breeding success. But when you look at the lifetime, it does seem like they are getting more offspring over their lives, and we think this is being driven by the effective lifespan that I showed on the last slide. That they're not necessarily better at reproducing, but they are better at living longer and reproducing over their whole lifespans. And maternal loss, which is the final plot on here, shows that individuals who are orphaned during their first year of life have reduced lifetime breeding success, and this is the same for males and females. And this is the first time we've really seen long-term effects of orphaning in this system, so we're not really quite sure why we're seeing this effect, although it is still really interesting. It could be that this sort of orphaning term is capturing something in the model which the other environmental terms aren't capturing, or it could be that individuals whose mothers die lose on some sort of like social or physiological function within the system. So to summarize quickly, harsh early life conditions do indeed have consequences for adult fitness and lifespan in this system, and the consequences are stronger for males, as we can see through the birth weight and population size. There are different processes contributing to this, so the classic silver spoon effects, but also this like selective filtering idea is coming through the effect of harsh winter weather on male lifespan. And I'd like to highlight that highlight the important use of using multiple aspects of the early life environment for wild populations. And I'd like to say that next I'm going to see how early life adversity might impact the onset and rate of senescence in the system, and whether that differs between the sexes. So thank you for listening. Thank you. Yes, thank you. Quickly, thank you to my supervisors and everybody who works on the project. I mean, I'm a computational project, and I can't do this work without the really important field work that goes on. Yes, thank you. Oh, and scan the QR code or email me if you want to find out about the manuscript. I'm going to put it up on bioRxiv soon, but haven't quite managed it yet. Okay, thank you very much.
Hi everyone, I'm Juliette Barbeau, a master's student at University of Sherbrooke in Quebec, and I'm glad to present to you today the first result of my study on the interplay between reproduction and telomere length variation in female tree swallows. So the notion of trade-off between life history trait is a key element of my study. Specifically, I'm focused on the trade-off that would exist between reproduction and survival. But this concept is a tricky one to observe in the wild study, where some individuals tend to have better access to resources or in better condition, which allow them to allocate a higher level of energy to both a higher reproductive performance and personal support. And they can then mitigate the cost of reproduction and would instead see this heterogeneity between the traits. Some study has observed this trade-off, like for the kestrel, where an artificially enlarged clutch is associated with lower survival, while others have observed this heterogeneity, like for lemurs, where higher litter size is associated with higher survival. And it's not improbable that an increased investment in reproduction could also have this layover costs on survival, which could not be immediately observed. And so to better understand how to quantify this trade-off, we could use molecular biomarkers such as telomeres, who serve the crucial function of protecting the integrity of eukaryotic chromosome. However, their position at the end of chromosome renders them vulnerable to replication problems during each cell division. They will then progressively shorten during the life of an organism until they reach a critical length threshold, which would trigger cellular senescence. Consequently, individuals with short telomere are widely recognized as having a shorter longevity. And because this process of shortening with age is accentuated with oxidative stress, for each age there is differences due to each's life experience. And so if there is this heterogeneity between life history traits, you would see that the variability in telomere length would both be associated with their survival and reproduction. And so individuals with longer telomere would have a better overall fitness and then would have a higher reproductive success for breeding activity. And it could also be associated with their individual variabilities, such as their body condition, which could be associated with their fitness. But in the lens of trade-off, reproduction could also represent a cost, and then this stress would have a cost then on telomere maintenance. And so a higher reproductive effort would accentuate the shortening of telomere due to a higher level of oxidative stress, and there will be this trade-off with future survival and possibly even future reproductive events. And so an individual telomere shortening could be used as a biomarker of their individual reproductive effort. Reproductive effort could be also difficult to measure in the wild study because for same reproductive output, like the number of eggs in a clutch, there will be a varying reproductive effort from one individual to another. If one is found in a higher quality environment, or if they are more experienced in reproduction, they could then minimize their telomere loss for same activity. But both theories aren't mutually exclusive, and you could both observe the heterogeneity and trade-off if you separate at the individual and population level. While looking at the variability of telomere length between individuals in a population, you would see that those with higher telomere, longer telomere, would have also a higher reproductive output, while at the individual level, you could see the trade-off if you repeat telomere length measured during the life of an individual. And so higher reproductive effort would generate more telomere shortening. And it's then essential to differentiate between these two levels. And so for my study, I examined the variability in telomere length between female tree swallow to really observe the telomere dynamic at the population level and see if those differences are due to individual differences such as their age and body condition, and if it can be associated with their reproductive success. So I expected that longer telomeres would be found in females who are younger, and for each age class would be in better body condition, like higher mass and longer wings, and also have a higher reproductive success, like higher number of fledglings. And then I examined the variability of changes in telomere length over a breeding season between the female to instead look at the telomere dynamic at the individual level and determine which factor will influence those variation in changes. So I expected that more telomere loss would be found in females who have better reproductive trait and success, like earlier length date, larger clutches, and a higher number of fledgling. But because we know that reproductive effort should vary, it should be accentuated in females who are younger and so less experienced in reproduction, and also found in lower quality environments with less available resources for them. And so my study model is the tree swallow, an insectivorous migratory passerine who breeds in North America from May to July and migrate to Mexico during the winter. On average, they have clutches of 5 to 6 eggs, but only fledge 3 to 4 chicks, and generally, reproductive trait will improve with a female age. And we can also identify the female age on the field, so we either have the second-year female who are 1 year old and it's their first year reproducing, or the after second-year female who are 2 year old or older. A shortening of telomere length has already been found in this species, where older individuals tend to have shorter telomeres, and both a trade-off and heterogeneity has been found between telomere length and reproductive trait. So a trade-off with the nestling mass and an heterogeneity with the laying date. Our study system is distributed on 20 farms in southern Quebec on over 10,000 kilometers square, and on each farm there can be found 10 available nest box for reproduction, which is completely monitored from the nest building to fledging, which allows us to have access to many different reproductive traits. We sample blood during two moments during the reproduction, so during an incubation period and during the post-hatching period, which allows us to have access to two different measures of relative telomere length per individual obtained by the qPCR method, and then gives us these changes in telomere length seasonally with an average of 12 days between the two captures. So for my first question, I look at the telomere length variability between the females, and so I did the linear mix effect model for each capture period, so incubation and post-hatching. First, I did the age as a binary variable of either the second year or after second year. then the mass and wing length as indicator of their body condition, and also the hour of capture and laying day to control for the temporal variability of the mass. Also the clutch size and number of fledgling as other reproductive trait, and 3 random effects, so the date of analysis, the ID of the plate for telomere analysis, and also the farm on which the female bred to consider the environmental differences between each site. And each fixed effect was also considered as an interaction with age. And so during the incubation period, we saw this effect of the interaction of wing length and age on telomere length. So only for the second-year female in red, we saw that longer wings was associated with longer telomere. And they also seem to have longer telomere than the after second year in blue, but only for those with better body condition with longer wings. And so while there seems to be this slight shortening with age and telomere length, for the younger age class, it's much more variable and seems to be dependable on this on wing length, so associated with their body condition. While for the after second year, if they survive up to this age, the variability in wing length will have not much of an impact on their variability in telomere length. And during the post-hatching period, we saw this effect of mass on telomere length, where female of higher mass also had a longer telomere, regardless of their age. And because mass is a morphological trait that is more susceptible to change during reproduction due to a higher reproductive effort, it seems to support why it's only associated with telomere length at the end of reproduction. Thus, it's the individual that retained the highest mass at the end of the reproduction that also had the longer telomeres. And also during the post-hatching period, we saw this effect of the interaction of number of fledgling in age, but it wasn't what was expected. And so again, the second year seems to have slightly longer telomeres than the after second year, except for those with a really high number of fledgling. And only for this age class, there was this trend where higher number of fledgling was associated with shorter telomeres. And for whatever reproductive success for the after second year seems to have much of an impact on their reproductive success, and so it could not be used as a biomarker of their reproductive success. Instead, for the second year, we see more of this trade-off at a population level, because for this age class, reproduction could represent higher costs because they are less experienced. Those who invest more in their reproduction, which are able to bring their young to fledgling, who results in having shorter telomeres. And so overall, we saw that the younger female have slightly longer telomere than the older one, but it's much more variable and dependable on other individual traits. And it seems to stabilize within the next generation, either due to individual shortening or for a population selection with age, which could be answered with a longitudinal study. And so for my second question, I look at the changes in telomere length variability. And so I did a linear model for each female that were captured at both periods. So the model was similar to the first one, but also added the changes in mass and Julian date between captured, and also the density of population on each farm as a proxy of environmental quality. So farm of higher density was considered of higher quality. And only the age of the female seems to have an impact on the changes in telomere length seasonally. So if the changes was 0, it means that it was constant, while a negative value means that there was shortening during reproduction. And it seems that older after second year female show greater telomere shortening than the younger second year female. So contrary to expectation, the experience acquired with age and reproduction does not help to minimize somatic costs. And these results seem more in line with the terminal investment theory. So the older you get, the more you need to invest in your current reproduction to your own detriment, because you will have less opportunity to reproduce in the future. And so while the after second year in our system have better overall reproductive success, it doesn't come without a personal cost under telomere maintenance. But we cannot ignore that we have a relatively small sample size, and we will replicate this on another breeding season, and instead look at the changes in telomere length between 2 breeding seasons to see more of this layover effect of the cost of reproduction. And so to conclude, we saw that the dynamic of telomere length in the female tree swallow is largely dependent on their age, while for the second year female, they seem to experience more of a trade-off at the population level with their number of fledgling. The after second year experience more of an individual loss in their telomere length. And so possibly telomere length could be used as a biomarker of the trade-off between life history trait, and could also be used to explain the variability that exists, particularly in the younger age class. And so I would like to thank everyone, my supervisor, everyone who contributed to the project, either on the field or in the lab. The farmer who gives us access to the land for now over 20 years, and also Claudiane Langlois, who's also here today. She did a poster on telomere, so you could also go talk with her about it. And thanks to you. I don't know if we have time for questions, but if anyone have question, I could take one maybe. No, okay. Oh, okay, sorry.
Do you have plans to assay genetic variation of any of the loci that are known to affect telomere length?
No, well, we're actually only using telomere length as a trait for now, but possibly what could be interesting is also looking at other maybe genes or structure that are implicated in telomere shortening to see how those are affected, but for now, only telomere length will be used. Thank you.